So this is the first of two lectures for Chapter 14 of Unit 3. In this first lecture here, we're going to talk about the citric acid cycle, and in the next lecture for Chapter 14, we're going to talk about the electron transport chain. So again, an overview, you have the citric acid cycle that takes place in the mitochondria, and then it functions to oxidize acetyl-CoA to carbon dioxide, yielding NADH and FADH2, those electron transporters. So you have a cell like this, and it's got a nucleus like this, and the cytosol's out here. And then you have the mitochondria like this. And you have that inner membrane right here and the outer membrane like this. Now remember, glycolysis takes place within the cytosol and produces pyruvate, which then travels to the mitochondria and then is converted into acetyl-CoA, which then enters the citric acid cycle. Which takes place in the mitochondria and then yields NADH and FADH2. And then these guys enter the electron transport chain, which is found in the inner membrane of the mitochondria, and it functions to carry out the process of oxidative phosphorylation. We'll explain what this term means in a second here. And then at the end of this, you produce ATP. So that's just a very general overview. Now we'll go through these steps in much more detail throughout the lecture. So the electron transport chain is a group of protein pumps, as you can see here, found in the inner membrane of the mitochondrion. So this is the inner membrane here. And they carry out a series of oxidation reduction reactions that transfer electrons from electron donors, NADH and FADH2, to an electron acceptor, which is oxygen, O2, the oxygen we breathe in through the lungs. And so as you can see through, just as an overview, the process is the electrons get transferred down from each of these complexes and then eventually get transferred to oxygen to form H2O here. Now, these redox reactions, they power the pumping of hydrogen from the mitochondrial matrix out here to the inner membrane space. So you're building up the concentration of hydrogen within the intermembrane space to create a concentration gradient or electrochemical gradient, where the concentration is much larger in the intermembrane space versus the mitochondrial matrix. And this electrochemical gradient is what drives the ATP synthesis, because you have this buildup and then eventually it comes back down through this protein called ATP synthase, and that powers the phosphorylation of ADP plus phosphate to ATP. So the term oxidative phosphorylation comes from where the oxidation of electron donors is coupled to the phosphorylation of ADP to synthesize ATP. And these processes are coupled via the movement of hydrogen ions across the inner membrane of the mitochondria. So the redox reactions drive hydrogen into the intermembrane space, create that gradient, and then that gradient drives the synthesis of ATP. Again, like we said, NADH and FADH2 are oxidized by electron transport of the mitochondria to generate ATP. Each molecule of acetyl-CoA yields 10 ATPs. Although two of the reactions of the citric acid cycle occur in the cytosol, as a whole, it's considered to occur in the mitochondria. And you can see that here. We show this green circle representing it. It's a major player for both anabolic and catabolic processes. Anabolic is building things up, and then catabolic is breaking things down. You could potentially remember because of anabolic steroids, which help build muscle mass. It's the same kind of thing. Building up, catabolic, breaking down. Glycolysis, for example, is a catabolic process because it's breaking glucose down, versus gluconeogenesis is an anabolic process because it's synthesizing glucose. The citric acid cycle contributes intermediates to the synthesis of glucose, fatty acids, sterols, porphyrins, urea, and the net interconversion of amino acids. So these intermediates can serve a lot of other purposes as well. It also oxidizes acetyl-CoA generated from the breakdown of glucose. So glucose is broken down to pyruvate and then to acetyl-CoA, which then enters the citric acid cycle, which then enters the electron transport chain to generate ATP. You can also have mobilization of triglycerides, where they are broken down into fatty acids and glycerol. Fatty acids can then be broken down to acetyl-CoA, which can then also enter the citric acid cycle. And then you can also have breakdown of proteins to amino acids, which then can be broken down to acetyl-CoA, which again enters the citric acid cycle. So acetyl-CoA, as we show here in this diagram, can come from multiple different sources. In major anabolic tissues such as the liver and the kidney cortex, the citric acid cycle is open. And what that means is that these intermediates, so each of these is an intermediate and so on, they can flow from the cycle, so they can flow out or they can flow in, and when they flow out, they can be used in other biosynthetic pathways. And as a result of that, the citric acid cycle will alter its rate of flux based on the energy needs of the cell. So if there's a greater need for energy and ATP, the citric acid cycle will increase its flux. If there's a decreased need for energy, obviously it'll decrease its flux. All right, so over the next few slides, we're going to go over each of these reactions within this diagram that illustrates the citric acid cycle. So it begins with pyruvate entering the mitochondria, and then it's converted to acetyl-CoA, and then is able to enter the citric acid cycle. So this conversion of pyruvate to acetyl-CoA is carried out by an enzyme called pyruvate dehydrogenase, key player in this process. It's an irreversible reaction, meaning that once you've committed to creating acetyl-CoA, you can't go back and synthesize pyruvate from acetyl-CoA. Now, the substrates for this reaction are pyruvate, then you need NAD+, CoASH, and then you also need 5 cofactors: thiamine pyrophosphate, or vitamin B1, lipoic acid, vitamin B5, FAD, and NAD+. And what this converts to is obviously acetyl-CoA, NADH, hydrogen, and then CO2. And so you can see you have an NADH here, and we indicate these in blue, and then FADH here, and then NADH here, because obviously these are important because these are the electron carriers that are then going to go to the electron transport chain where you produce ADP. Now, what's important in biochemistry is the regulation. Why does certain reactions proceed? Why do some do not? What certain state is the cell or the body's physiology in that causes those certain reactions to go faster rate or to be inhibited? So, for example, you have NAD+, ADP, and then calcium. All 3 of these activate pyruvate dehydrogenase. That makes sense. You have a buildup of NAD+, you're going to want to push this reaction forward. Buildup of ADP means that you have a lot of ADP that's ready to be formed into ATP, but to get it there, you got to end up generating these electron carriers so that the electron transport chain can carry out its function. So a buildup of ADP is going to push this reaction because it's going to mean that the cell is depleted of energy and is ready for new energy generation. And then calcium also activates this as well. There's also feedback inhibition. Remember, when there's a buildup of a product, it comes back and inhibits the enzyme that synthesizes it to help prevent overproduction. So you have acetyl-CoA that comes back and negative inhibits. And then you have NADH, which also comes back. Because at a certain point, if you have a buildup of this, you've kind of reached a saturation point. You don't necessarily need to synthesize any more of this. That's the point you reach where you come back and feedback inhibit. So you have acetyl-CoA, you've synthesized that, and then it enters the citric acid cycle by combining with oxaloacetate, and that forms citrate. And this reaction is catalyzed by citrate synthase. Now, citrate synthase receives negative feedback from citrate, a competitive inhibitor, which means it binds at the active site of the enzyme. So it comes back and inhibits citrate synthase. The other thing that's important to note is that when you have a buildup of ATP, this comes back and is a negative allosteric regulator, meaning allosteric meaning it binds to a site on the enzyme other than the active site. And remember, the active site is the site on the enzyme protein structure where it's carrying out its function of catalyzing the reaction. Anyways, you have ATP come back, negative inhibit, and what that does is that it decreases citrate formation during periods of energy generation. So you're generating a bunch of ATP. You've also reached a point where it's diminishing returns to produce any more of these NADH and FADH2 at that point because you're producing a high number of ATP and producing energy. So as a result of decreasing citrate formation, you decrease citric acid flux. And so the citric acid cycle is running at a decreased rate because you're in a state of high energy generation. So the next step in the cycle is a lower yield step, so we'll just kind of briefly mention it. It's where you go from citrate to isocitrate, and this is catalyzed by aconitase. And really that's all you really need to know about this. I guess also it's a 2-part reaction. Again, not a very particularly high yield step to know. Next, you have isocitrate, which is this intermediate produced. And this is error in your book. Make sure you just cross this out, dehydrogenase. We apologize by that. We're in this sentence, we're really just referring to the intermediate. We're not referring to the enzyme doesn't undergo decarboxylation reactions. It's the intermediate. So isocitrate then undergoes two oxidative decarboxylation reactions, meaning they're losing CO2 in each step here. So it's both these two reactions here to form succinyl-CoA. And as a result of these two reactions, you produce two equivalents of NADH as well from each reaction. So the enzymes for each of these reactions, so first, the first reaction, you have isocitrate dehydrogenase, catalyzes the formation of alpha-ketoglutarate from isocitrate. Now again, as we've talked about earlier, is that these enzymes are often particularly sensitive to the energy levels in the cell. So if there's high levels of ADP, meaning that you have lower levels of ATP, and you're in a lower energy state, this is going to be an allosteric activator. So it's going to help push this reaction forward because you want to produce NADH, and you want to push the cycle forward and end up producing more ATP. However, on the flip side, it's also sensitive to high levels of NADH. So this NADH will come essentially, you can think of it as feedback inhibit, or being sensitive to high NADH levels will feedback inhibit the enzyme. Also, if you've produced high levels of ATP, high energy state, the citric acid cycle isn't under as much pressure to push out NADH to produce ATP. So if you have high levels of ATP, that also comes back and negative inhibits as well. So it's a negative allosteric regulator. Alpha-ketoglutarate dehydrogenase, this converts alpha-ketoglutarate to succinyl-CoA. And this is the second reaction of these two reactions. So what this does is it adds a S-CoA group using CoA-SH as one of the reactants. And then it also is a decarboxylation reaction by taking off a CO2. This one, again, it produces an NADH, but it's also sensitive to high levels of NADH. So high levels of NADH will come back and feedback inhibit. Again, just like the previous reaction, if you have high levels of ATP, this is going to be a negative allosteric regulator as well. The other thing is if levels of CoA-SH are low, this is going to decrease the rate of the reaction. And because it's one of the reactants, so if you have a lower concentration of one of the reactants, it's going to lower the rate of the overall reaction. And then succinyl-CoA is also going to feedback inhibit onto this enzyme as well. Lastly, it's important to know for some of these enzymes, they require vitamins as cofactors. And so alpha-ketoglutarate dehydrogenase requires vitamin B1, B2, B3, B5, and lipoic acid. All right, so for the next step in the cycle, you have succinyl-CoA, which is then converted to succinate by the enzyme succinyl-CoA synthetase. This reaction carries out what's called substrate-level phosphorylation. Traditionally, you think of producing ATP, you take an ADP and then an inorganic phosphate, and together that produces ATP. In this case, you take high energy substrate and then convert it to a lower energy substrate, and typically that involves a substrate with a phosphate attached to it, and you basically steal that phosphate and transfer it onto, in this case, a GDP to then form a GTP. So that's the difference for what substrate level phosphorylation is versus more traditional thought of how ATP is produced. The other thing is that GTP feedback inhibits on this enzyme. So if you're producing high levels of GTP, it's going to feedback inhibit. The other thing is this GTP is eventually converted to an ATP. So one turn right there, we've already produced an ATP molecule from the citric acid cycle. Then you have succinate dehydrogenase, which catalyzes the oxidation reduction reaction that converts succinate into fumarate here. This yields an FADH2, which is very important to know. Again, just like NADH in these reactions, if you have a buildup of FADH2, this will feedback inhibit on this enzyme and decrease enzyme activity. And then next we have fumarate. This is kind of a lower yield step, and really just to know is that the enzyme involved is fumarase, and it converts fumarate into malate. And this is a hydration reaction, meaning that you've added water in, and that's really all you need to know about that reaction there. All right, so this final step here consists of malate dehydrogenase catalyzing the formation of oxaloacetate from malate here, and this yields an NADH. Now this is a reversible reaction, which means that it can actually go either way. And the equilibrium actually favors the malate formation. So it actually favors going backwards in a sense in terms of the citric acid cycle. Now you may wonder, well, then how do you move the citric acid cycle forward? How do we get to oxaloacetate? The flux of this reaction in reality, in normal physiology, actually favors oxaloacetate formation because oxaloacetate and NADH are rapidly removed by other enzymes in the mitochondria for other important reactions that we'll talk about in subsequent lectures. So as you're forming these, they're rapidly being removed and rapidly the concentrations being decreased. And so that's going to help favor this reaction continuing to move forward in this direction. Because if you're decreasing these end products, that's going to drive the equilibrium that way and drive the reaction that way. Now, just like some of these other reactions, if you have increased levels of NADH, if this does happen, comes back, inhibits and decreases the activity of the enzyme. Another thing that occurs in this situation is that malate accumulates, and this is actually important during gluconeogenesis, which we'll talk more in detail about in the gluconeogenesis lecture. And just NADH levels have a huge influence on the flux of gluconeogenesis. So that's the last step, and then when you have another acetyl-CoA coming in, that can join with oxaloacetate and then form citrate and then continue the process forward. So as far as the citric acid cycle products, I do apologize. I would just cross this out here. This is incorrect. I want to make this much more clear and walk through this with you. This fact is true. One glucose molecule produces 2 acetyl-CoA, and that's important to know. We'll talk about that in a second. So let's count up these products. Yes, you do produce 2 CO2, but that's not as important as you have 1, 2, 3, 4 NADH. So you have 4 NADH. Now, like I said, one glucose molecule produces 2 acetyl-CoA. So you end up getting twice of this because you produced 2 of these. So you're going to do 2 rounds of the citric acid cycle because for each of these. So that'll be times 2. So you end up producing 8 of these. And then you have 1 FADH2, which is produced during this. And again, times 2. So that will give you, obviously, 2 FADH2s as a result. Now, what's really key here is how much ATP results from this. So remember, you get this GTP here, and you're going to end up getting 2 GTPs, and these both end up getting converted into 2 ATPs. So right off the bat, you start with those. And then these are going to go to the electron transport chain and get converted into ATP. And so for each NADH, you actually get 3 ATP molecules. So times 3. So you get 24 ATP from the NADH. And then for each FADH2, you get 2 ATP. So times 2. And you get 4 ATP. So for the citric acid cycle, you end up getting 30 total ATP. Now, but you got to remember with glycolysis, and again, we'll go over this in more detail in the glycolysis lecture, but glycolysis, you actually produce 2 net ATP, so you start out with that, and then you also produce 2 NADHs, which then can go to the electron transport chain. Remember, that's times 3, and so that'll produce 6 ATP. So glycolysis is producing 8 ATP, so 30. So then down here you have the 8 from glycolysis plus the 30 from the citric acid cycle, 38 ATP total. And this is theoretically, this is not necessarily going to be producing 38 every single time. There's other circumstances in the cell that can affect the final yield. But for purposes of exams, purposes of studying this, you want to think of theoretically you would produce 38 ATP per 1 molecule of glucose going through the entire aerobic respiration, meaning going through all of glycolysis and then going through the citric acid cycle. And so when they talk about the final products of citric acid cycle, that's what's important is that you produce 4 NADHs, 1 FADH2, and then you have 2 ATP, and then you get that 2 CO2 as well. All right, so over the next couple slides, we're going to talk about cataplerotic and anaplerotic reactions. This isn't terribly high yield for the USMLE board exams, but which still could come up on a biochem exam and could be covered in a biochem lecture in your course, so we're just going to touch on it briefly here. So cataplerotic reactions, they consist of the outflow of intermediates, so intermediates coming out from the citric acid cycle to be used in other biosynthetic or metabolic reactions. So these are important intermediates that can be used elsewhere as well. So those would be cataplerotic reactions that would essentially take those out. And then you have anaplerotic reactions, which essentially refill those intermediates for the citric acid cycle in the liver specifically. So these reactions happen in the liver. And these are 4 examples of those, and we'll go through each of those over the next few slides. So first you have for replenishing oxaloacetate, you can take pyruvate, combine it with CO2 and ATP. This is catalyzed by pyruvate carboxylase, and this enzyme requires biotin, also known as vitamin B7. Acetyl-CoA is a positive allosteric regulator. So if you have a buildup of this, you're able to convert this if you add in CO2 and ATP. This would be pyruvate carboxylase. So if you have a buildup of this, it stimulates this conversion here to help you replenish oxaloacetate, which then can combine with acetyl-CoA to then form citrate and get the cycle started. You can also form oxaloacetate by combining aspartate with alpha-ketoglutarate, catalyzed by an enzyme called aspartate aminotransferase, which requires vitamin B6. Aminotransferases, as we'll talk in much more detail about in the amino acid metabolism lectures, are very important for amino acid metabolism, but just you don't need to know anything really beyond that at this point, but we'll go in more depth on those in those lectures, but just to make you aware of that. Also produces a glutamate, and then this glutamate is used in the following reactions. So you combine it with an NAD, and then you yield a alpha-ketoglutarate and an NADH, and then ammonia as well. And this is catalyzed by glutamate dehydrogenase. Here you kind of have 2 reactions that work together to eventually give you succinyl-CoA. So you start with propanil-CoA plus CO2 plus ATP, gives you methyl-malonyl-CoA. And this is catalyzed by propanil-CoA carboxylase, and this requires vitamin B and biotin. Then methyl-malonyl-CoA is converted to succinyl-CoA by the enzyme methylmalonyl-CoA mutase. And I believe in your book it doesn't have mutase written there. That's an error. Make sure you add that in. We apologize about that typo there. So it's methylmalonyl-CoA mutase is the enzyme, and this enzyme requires vitamin B12 to function. And so that helps give you succinyl-CoA. All right, so that wraps up our discussion of the citric acid cycle. In the next lecture, we're going to cover the electron transport chain.